Hi everyone, this is Dr. Fox, and let's talk about nephrons. In this video, we'll describe the nephron and its features, as well as the processes of glomerular filtration, tubular secretion, and reabsorption. We'll detail the substances that are secreted and reabsorbed in various parts of the nephron, explain how diuretics affect urine formation, and explain the differences between cortical and juxtamedullary nephrons. So let's start with the functional unit of the kidney, the nephron. The nephron, as a structure, filters blood such that we can make urine. There are two important portions of the nephron, the renal corpuscle and the tubules. The renal corpuscle has two distinct parts, the glomerulus, which is a specialized capillary bed, which is surrounded by the glomerular capsule. The glomerulus is fed by an afferent arteriole, and it is drained by an efferent arteriole. And the efferent arteriole goes into a system of peritubular capillaries that surround the tubules. These peritubular capillaries and the tubules have two distinct processes that are occurring. They are tubular secretion and tubular reabsorption. Now, we can pair this along with glomerular filtration. So, glomerular filtration is the process of converting blood into a substance known as filtrate. Then the processes of tubular secretion and reabsorption refine that filtrate such that at the end of these processes, we're left with urine. Now, these processes are going to be very dynamic, and they are going to depend on conditions within the body, such that anytime we need to move materials from the peritubular capillaries into the tubules, we call that tubular secretion. And anytime we need to reclaim or reabsorb materials from the filtrate back into the cardiovascular system, we call this tubular reabsorption. Now, at the end of the tubules, these feed into structures known as collecting ducts. And generally, multiple nephrons are going to feed their filtrate into collecting ducts. And those same processes of tubular secretion and reabsorption will continue in those collecting ducts. So let's begin with the renal corpuscle. The process that occurs at the renal corpuscle is glomerular filtration, and this is just the process of converting blood from within the glomerulus to filtrate, which is collected by the capsule. So we have an afferent arteriole which feeds blood in. This blood is filtered into filtrate, and that filtrate moves into the tubules. Now, this capsule is a serous membrane-like structure in that there is a parietal portion which covers the walls and a visceral portion which covers the glomerulus. Now, it is only like a serous membrane. It is not true serosa. So let's look at the visceral layer, or the filtration membrane. The filtration membrane consists of a layer of cells called podocytes. And podo means foot. Basically, these podocytes are going to put out these little tiny feet which interdigitate with other podocytes that surround the glomerulus, such that there is gap between them, and these gaps are known as filtration slits. So we create a filter. Now, helping or aiding the podocytes are mesangial cells. Mesangial cells are smooth muscle tissue that sit between the glomerular loops, and as they contract, they pull the podocytes closer together, and as they relax, they allow them to drift further apart. So we are able to fine-tune glomerular filtration rate. Generally, however, GFR, glomerular filtration rate, is going to be proportional to blood pressure. So as blood pressure increases, so does glomerular filtration rate. As the flow of blood increases, so as afferent arterioles dilate or get larger, glomerular filtration rate increases, as we'll see. So filtrate moves from the corpuscle into the tubules, and the first part of the tubule is known as the proximal convoluted tubule. Recall that proximal is closer to the beginning of a tube and distal is further from the beginning. So the proximal convoluted tubule is the first part of the tubule, and this is the primary site of tubular reabsorption. This cannot be understated. And so when we think about all the materials that are reabsorbed here, we have salts and water which follow the salts. So about 2/3 of the final reabsorbed volume occurs here in the proximal convoluted tubule for salts and water. Also, glucose and amino acids. Renal clearance of glucose in a healthy individual is 0%, meaning that 100% of the glucose and amino acids get reabsorbed. They are not cleared from the filtrate into the urine. We also have potassium being reabsorbed here, phosphate being reabsorbed here, citrate being reabsorbed here, some urea being reabsorbed here. About half the total amount of urea is reabsorbed here. And we have a number of substances which are secreted. So ammonia, as a waste product of glucose catabolism, is secreted from the peritubular capillaries into the proximal convoluted tubule, as well as any bioactive substances, pharmaceuticals, etc. Now, the proximal convoluted tubule has a region which is rather straight and a region which is rather curvy or convoluted. And we call these the pars recta. Recta is straight, so that's the straight part, versus the pars convoluta, or the convoluted part. The proximal convoluted tubule is going to feed the filtrate into the nephron loop. So we go from the proximal convoluted tubule into the nephron loop. The role of the nephron loop is to further reabsorb water. Now, the proximal convoluted tubule may be the primary site of water reabsorption, but the nephron loop, or the loop of Henle, is going to help with this. And it does so by creating a hypertonic medulla. Now, the loop of Henle is firmly anchored within the medulla, and water is always going to move from a lower to a higher tonicity, or toward a hypertonic environment. And how this occurs is that because this is a hypertonic environment, water is going to flow out. But first, sodium is reabsorbed passively, and then sodium is reabsorbed actively. So we have active sodium transporters there. And the end result of this is the hypertonic medulla. Water follows sodium, and that excess water gets pulled by specialized vessels called vasa recta, which reabsorb that water from the nephron loop back into the cardiovascular system, leaving the sodium to further reinforce that hypertonic medulla. Now, from the nephron loop, filtrate is going to move into the distal convoluted tubule. The distal convoluted tubule has a variety of cell types. There are intercalated cells, and the role of intercalated cells is to help fine-tune pH. So, for instance, they are capable of secreting or reabsorbing bicarbonate ion or protons. There are two different types cells within the intercalated cells that can keep or excrete either of these ions. These intercalated cells are also present in collecting ducts, so that work can continue after the filtrate leaves the nephron. There are also principal cells, and the role of principal cells is to help with the reabsorption of sodium, and following sodium comes the reabsorption of water. This is something which is aided by the RAA, or the renin-angiotensin-aldosterone system. And also, principal cells are capable of reabsorbing water with the secretion of vasopressin, or antidiuretic hormone. Other epithelial cells within the distal convoluted tubule are capable of reabsorbing calcium, and this process is modulated by parathyroid hormone. So you may recall that parathyroid hormone helps to modulate calcium homeostasis, and one of the ways it does so is by aiding the reabsorption of calcium in the distal convoluted tubule. Now, those intercalated cells and principal cells are also found in collecting ducts, and so their role will continue after filtrate moves from the nephron and into a collecting duct. There is also a specialized cluster of epithelium within the distal convoluted tubule that is adjacent to the glomerulus, which is called the macula densa. The macula densa is going to be a salt sensor. So this is going to detect the concentration of salts in the filtrate, and this will serve as a proxy for glomerular filtration rate, as we will see. So there are two distinct types of nephrons: cortical nephrons and juxtamedullary nephrons. Cortical nephrons are the most common type of nephron. They sit well within the cortex, which is why we call them cortical nephrons, and they have very short nephron loops that just barely make it into the medulla. We can contrast this with juxtamedullary nephrons. Juxtamedullary nephrons are much less common in humans. They are more common in desert-dwelling organisms where further refining water reabsorption is more important. Their corpuscles sit adjacent to the medulla. That's why we call them juxtamedullary, so against the medulla. And they tend to have larger glomeruli, which allow them to have higher glomerular filtration rates. They also have longer nephron loops, and these longer nephron loops are what allow them to further concentrate their urine. And it's with these long nephron loops that we have those vasa recta, or the straight vessels, that help to return the water back into the peritubular capillaries and cardiovascular system, such that we can pull out more water and maintain that more hypertonic environment. Now, diuretics. You may have heard of these. Diuretics are bioactive substances which can either increase the tubular secretion of water or decrease the tubular reabsorption of water. So here is the peritubular capillary, here is the tubule, and so we can increase tubular secretion, so we can move more water into the filtrate, or we can decrease tubular reabsorption, so we can not move as much water back to the cardiovascular system. The net result of this is diuresis, or making more water. Diuretics are one of the frontline treatments for hypertension or high blood pressure, and this is because they can very easily reduce blood volume via diuresis. A couple of examples of diuretics. Caffeine. So caffeine works a number of ways. It increases heart rate, but it also slows water resorption. And by increasing heart rate, it also increases glomerular filtration rate, although all of the mechanisms for that aren't yet fully known. Another example of a diuretic is alcohol. Alcohol inhibits the secretion of vasopressin, or antidiuretic hormone. Vasopressin, or antidiuretic hormone, is really important for the reabsorption of water in the distal convoluted tubule, and such that alcohol consumption is going to slow water reabsorption. So if you have ever had the experience of consuming alcohol and then feeling as though you needed to micturate or urinate, that is because you are losing water volume from your cardiovascular system to your filtrate. And it is greater by the mere consumption of alcohol and by the volume that you are consuming. Now, if you're going to double down on an alcoholic drink that also contains caffeine, so vodka Red Bull, for instance, you are laying a diuretic on another diuretic. So please do be careful with that. And if you are out consuming alcohol, please do make sure to hydrate. Intersperse water here or there to maintain your health. And your body will thank you the next day, I promise. So let's assess the knowledge. Where in the nephron is the primary site of water reabsorption? The glomerulus? Nope, the glomerulus is all about filtration. The proximal convoluted tubule? Yes, about 2/3 of water reabsorption happens there. That is the principal site. Nephron loop definitely helps. Distal convoluted tubule definitely helps. Collecting ducts can definitely help, but none are going to compare to the proximal convoluted tubule. Thank you so much for your time, and I look forward to talking to you in the next video.